Thank you for. I would like to start with thanking to the organizers for this stimulating meeting. It's great to see there are still some people staying at the last, one of the last sessions of the conference. I just want to make a disclaimer: there is no deep learning in this talk, so if you want to leave, it's perfectly okay. So today I will going to talk about a work that we published recently, and this is a joint work with Dr. Menche from SEM in Vienna. And basically, when we think about cells, the cells are governed by many biological processors, so-called pathways. And when we have some perturbations in pathways, this gives rise to certain diseases, right? In certain pathways, mutation in certain pathways gives rise to diseases. And we would like to find the signatures for diseases. And the way we do this usually is looking at samples, grouping them as healthy and disease. But oftentimes, this is actually a strong assumption because we know that most of the time, disease phenotypes are not clear-cut, and there are many subtypes. And this has important implications in the clinic. So if you look at, for instance, atorvastatin, one of the most successful drugs in the field of cardiovascular diseases, there are many clinical trials associated with it. And if you look, for instance, into one of them, where they look at almost 600 patients, they treat them with the drug, and after 6 months they check, and almost half of the patients, they don't really meet the clinical endpoint, meaning that there's a huge patient-level heterogeneity in terms of drug response. So we turn our attention to transcriptomics data to be able to quantify such kind of patient-level heterogeneity, right? And this is an example where we have in asthma some gene expression data, and we look at the expression values of a known asthma biomarker, periostin. And if you look at the expression levels of this gene, you would see that there are many case subjects, asthmatic subjects, that they tend to have an expression of this gene lower than the control subjects, the healthy individuals. So although this is a biomarker, it's not necessarily displayed in all the samples. And we know this, especially in cancer, this is already known that the driver genes, they're not in all samples, so we know that like only in few of genes are produced in each sample, but like we don't really have a way to quantify this sort of information. To this, if you generalize this and if you look at the genes that we identify by a group-wise differential expression test, like say you use a t-test, Lima, whatever package that you like, you would still see that there are many genes that are in the differential express set, but they don't appear in individual samples. To take into account this kind of effects, we develop a very simple framework. So we try to find in each individual the genes that are specifically perturbed in that individual. And to do this, we use the gene expression, distribution of gene expression across control individuals, the healthy individuals, and then we find which genes in a given patient, it could be a case or control sample, falls in the extreme of this expression distribution. We call this profile as the personalized expression profile or personalized perturbation profile, if you wish. And these are the genes that are significantly perturbed in each individual. So the first observation, which is somewhat confirming the heterogeneity across patients, is that many group-wise differentially expressed genes, you don't really see them in the personalized profile. So most perturbations are specific to the patient. And if you look within the patients, like between asthmatic, or in this case we use 3 diseases, asthma, Huntington, and Parkinson's disease, if you look at the perturbation profiles within the individuals of the same disease, you see that there's still quite low overlap. So again, many perturbations are specific to the patient, but still this overlap is higher than what you see between healthy individuals, which is a bit reassuring because in the end, these signatures are characterizing the disease rather than some physiology of the samples, let's say. And next question is basically, are these perturbations falling under the same biological pathways? And when we look at it, we see that actually, yes, that there are in almost all the individuals, the pathways involved in the disease, they contain this individual perturbation profiles. But again, most of these perturbations are somewhat mutually exclusive. So they are falling to the same pathway, but like they are covering different parts of the pathway, right? And here, in this case, is an example of cytokine-induced inflammatory signaling in asthma, and we see that there are many genes that are falling to, that are covered in different samples. So we see that there are certain genes that are describing the individualized perturbation in each patient. Then the next idea is, can we actually leverage this to be able to say something about the disease status of samples, right? The idea is here that since there are like parts of pathways that are involved in disease, can we generate a big pool that contains this certain number of these genes, and then use this pool as a gold standard. When you have a new sample, you can compare to this pool and see if you cover enough number of genes in this pool, and you can make predictions. But to do this, we have to make sure that we don't have any artifacts coming from biological or technical noise, which we make sure by normalizing, preprocessing the expression data. But still, we also have to make sure that like the disease heterogeneity is captured within the pools that we are going to define by number of common samples that we observe a given perturbation profile. To this end, we look at the number of genes that are shared in different samples, and we compare this information coming from the data with a random expectation that we calculate based on a very simple binomial model. So what is the chance of getting X number of genes common in two samples, and you can sum up these probabilities, and then you see that this is, the expectation is the green one, the random one, which is very consistent in the case of controls with the observation. And in the cases, we see that the number of shared genes are significantly higher than what you would expect by chance. So it means that actually, again, based on the number of samples you have, you can define a disease pool. And for this, I'm not going to get into details, but like we basically look at the maximum number of patients that a gene is expected to appear under a binomial model, and then we use this number as the threshold to define the size of the pool that we want to generate. And we optimize for this number, also the Z threshold that we use to define PEEPs, and then we show that we can predict the disease status of each sample to a fairly remarkable accuracy. In conclusion, we observe a high heterogeneity across patients at the transcription level, and we have developed a platform where we can use now this personalized individual perturbation profiles or expression profiles to quantify this heterogeneity, to define a pool that defines the disease, and use this pool to predict the disease status. There is an R package available online, and I'm going to thank all these funding resources that made this work possible, also the co-authors of the study, without whom the study would not be, of course, possible, from Northeastern University, Norik Santoni and Ellen Karma for their suggestions on the work, and to you for your attention. I will be happy then, more than happy to take your questions.
Thank you, Emre. Thanks for staying in your time. Please.
Interesting talk, thanks. So from a methodological statistical perspective, how do you get away from, biological replication and biological replicates? You only have one patient. How do you do that?
We don't handle we don't have replicates for each sample, basically, that's true, and we don't deal with it. But we try to make sure that we use the standard pipeline that everyone uses in the gene expression analysis, so that like the data set or initial data set that we use looks good enough to discriminate,
But how do you assess the significance?
And the significance in terms of the peeps, like the genes. I mean, this is a z-score, so basically you can convert it if you wish to a p-value, and you can set a p-value cutoff, or you can also set a z-score cutoff, basically, to set a significance. And you can also apply a false discovery rate correction, for instance, if you wish. Yeah.
Thank you, Emre.